Well, okay.
Could you able to see my slides right now?
We can, and then if you can hit like the little projector screen at the bottom.
Yeah.
Mhm.
Yeah, that's the one. Click it.
Yeah.
Looks good.
Perfect.
Yeah. Okay. So, thanks for the nice introduction, Dr. Eng. And today I would like to talk about translational research, what we are doing so far for the past 5 years. And the main target of today's work is phenyl diphosphate synthase, which is a key enzyme in the cholesterol biosynthesis pathway, and how we are going to show that as a target for radiosensitization, specifically for prostate cancer. That's what is going to follow later. So basically what I'm going to give, I'm not going to give much introduction about prostate cancer or radiation therapy because of this forum, which is already talked about SBRT or brachytherapy or some systemic therapy. But I would like to give some of the outlines or objectives and hypothesis, and what happens if you knock out FDPS or overexpress these FDPS, and how it could be targeted using a FDA-approved drug. And finally, I'll go with my summary and conclusion and future direction. So as we know that prostate cancer incidence, still it is estimated to be the #1 cancer in males. But when it goes for the death ratio, it is ranks #2 next to the lung cancer. So, but still it requires some more therapy interventions to reduce this mortality ratio in male prostate cancer. So what are the treatment options for these prostate cancer patients? As you know that these localized and locally advanced prostate cancer, they can be put in watchful waiting, active surveillance, and even the surgery, the radical prostatectomy. But mainly patients prefer to go for radiotherapy. And the radiotherapy is not only with the conventional RT, with the IMRT or brachytherapy, and nowadays it is coming up with the stereotactic guided radiotherapy as one of the main modality to treat these patients. A recent paper published from a European group showed that around 49% of patients, they prefer to go for surgery, and equally like around 40% of patients, they also prefer to go for radiation. So this makes that it as a major treatment modality, mainly in the low risk and intermediate risk and high risk patients, especially for locally advanced prostate cancer patient. When it comes to the radiation, we remember something that is called the radio resistance, and which exists because of the fractionated doses that makes it amplifies or enriches these radio resistant cells to acquire. Later, it helps for the prostate cancer to get recurrence. So what are the key issues with regard to the radiation, specifically in the localized prostate cancer patient? We know that when we radiate the cancer cells, the DNA damage signaling is evolving, and it can have a chance of giving rise to 5 of the phenotype. Either it can goes to apoptosis or necrosis or senescence, or it can induce autophagy, or it can go for DNA damage repair. But when it goes for autophagy or necrosis, definitely the cell will go for cell death. But when it goes for senescence or autophagy or DNA damage repair, the cell will go for cell survival, that is called the acquired radio resistance. So by this way, the cell will explore these mechanisms and take this as an advantage for the survival. And on occasion of these radiation induction or the radio resistance, there are certain molecules that overexpress, and that was shown to contribute for this radio resistance phenomenon. So hence, the overall conclusion of this slide is to make an understanding what are the mechanism which is involved in the prostate cancer radio resistance, and how to develop a novel strategies to target these specific molecules that are involved in the radio resistance. So initially we started this work basically from our prostate cancer work. One of our colleague, Michael Bain, Josh, in our lab, they initiated this work in pancreatic cancer. What they did, they radiated cancer cells to radiation, and they found that there are several differentially expressed genes that were evolved because of this radiation, and many of those genes were related with cholesterol biosynthesis pathway. So they conclude that this cholesterol biosynthesis pathway genes could be a target for radiosensitization. And they found that FDPS is one of the top gene which came in their microarray analysis. And they also did a tissue immunohistory analysis with this FDPS, and they found that the normal cell do express FDPS, but the cancer cells do express an increase in a trend of this FDPS expression. Surprisingly, the radiation-treated cancer cells do express much more higher level compared to the cancer cells even, stating that this FDPS is enriched or increased in response to radiation. So before going to the status of this FDPS in prostate cancer, I would just want to give a glimpse about this FDPS. So FDPS, we know that this cholesterol biosynthesis pathway starts with acetyl-CoA all the way to the mevalonate, squalene, and to the cholesterol biosynthesis. But FDPS is one of the key enzymes which is required and which is involved in the production of geranyl pyrophosphate and phenyl pyrophosphate from isopentenyl pyrophosphate and dimethyl halide pyrophosphate. So by doing so, we know that by the name by itself, it says that this FDPS is involved in the phenylation and geranylisation, which are all the post-translation modification required for the transfer or getting a hydrophobic moiety is getting attached to the substrate so that it can anchor to the cell membrane. These anchored cell membrane molecules can help for the protein-protein interaction or further signaling. So FDPS is not only involved in the CBS pathway, it is also involved in the post-translation modification. On the other hand, there are certain papers which are talking about the involvement of these nitrogen-containing bisphosphonates getting involved with ligand formation with human phenyl pyrophosphate synthase, and especially this R ligand that is present in all the nitrogen-containing bisphosphates has an ability to form a tri-ligand by forming a ligand with this magnesium ion present in the active sites of this human phenyl diphosphate synthase enzyme. And there is an equal chance of these nitrogen-containing bisphosphonate to form a ligand with the calcium ion present in the hydroxyapatite in the bone as well. So our rationale to find in FDPS as a target for this radiosensitization is by 4 reasons. We know that for the steroid hormone synthesis, specifically like androgens, cholesterol biosynthesis pathways involved, and the epidemiological studies showed that even an increase in 10 milligram of deciliter concentration of this cholesterol is associated with 9% chance of prostate cancer recurrence. And there is a paper which talks about the altered expression of this FDPS in prostate cancer, and they correlated this increase in FDPS level versus the Gleason score. A recent paper came in a cancer research and talking about global transcriptomic analysis in a formalin fixed prostate cancer tissues, and they identified like around 24 gene signature panels have shown to be elevated in these prostate cancer, and they were shown to be associated with the Gleason score as well. And among those 24 gene signature, FDPS is one among the top 5 panel of genes which was shown to be elevated and which was shown to be associated with the Gleason pattern as well. So, in the nutshell, we are rationalizing based on the high-risk disease patient, which is around 30 to 45 patients. They have a chance to get recur in the first 10 years of the radiation therapy. and among which 65% of patients have a local failure. This is mainly because of the radio resistant phenomenon. And we previously we identified this FDPS as one of the oncogene that is involved in the initiation and progression of prostate cancer. And our global gene analysis also shows that FDPS and this cholesterol biosynthesis pathway differentially responded against the radiation therapy. So based on this, we hypothesize that radiation by itself can enhance FDPS expression, which will positively modulate some intracellular signaling for getting the survival and growth advantage. Thereby, it increases the radio resistance. Thereby, we think that inhibition of these FDPS will enhance the radio sensitivity of prostate cancer. To prove our hypothesis, we basically had three main objectives: to understand the preclinical and clinical importance of these FDPS, and to determine the mechanism by which FDPS promotes prostate cancer progression and radio resistance. And we want to evaluate this in a preclinical model by targeting this FDPS using the radiosensitizer that we identified as zoledronic acid. So in the first objective, what we did, we surveyed the FDPS expression from a normal prostate all the way to the hyperplasia to the pin lesion, and we found that specifically FDPS is overexpressed in prostate adenocarcinoma as compared to the precursor lesion. Hence, we observed an increase in trend in this FDPS expression in the stage-wise, and we also looked in the in silico analysis. We found that the FDPS gene level is higher in prostate cancer, and with increase in the Gleason score, we could able to see a significant increase in the FDPS level with relative to the Gleason score 5 and 6. Apart from this, we also found a gene set enrichment analysis where we compared the FDPS level with the primary versus the metastatic, and we found that the metastatic prostate cancer tissues do express even higher level compared to the primary tissues, and this was verified in a coastal blown and a lymph node metastatic tissues as well. So based on this, we developed a mice model, and this is a traditionally used mice model for prostate cancer. It's a well-established mice model. That's a PETA knockout mice model, and we want to see whether FDPS has an association with PETA and whether it is associated with the progression of the disease as well. So in this one, we surveyed this FDPS expression, and we found that FDPS is highly expressed in all the lobes, dorsal lobes, lateral lobes, and this dorsal lateral lobes is the one which is similar for the peripheral zone that we are seeing in case of humans, and even it is expressed in the ventral zone as well. So that's the composite score, whatever we evaluated, it showed a high expression for FDPS in all the cancer tissues which we evaluated. And even though the organoids which was developed from this mice model do express a higher level of FDPS expression, showing that this FDPS is activated in PETA knockout model, and it could be associated with the oncogenic transformation as well. So based on this, our initial idea is to evaluate the FDPS expression both at the protein and RNA level, and we categorize these cell lines as a PETA wild type versus the PETA knockout or PETA functionally depleted condition. In this case, what we found, FDPS is expressed in majority of the prostate cancer relative to the normal immortalized cell line, but at the same time, this FDPS expression correlates with increase in the level of phospho-AKT as well. When we had an advantage of this PETA knockout cell line, mouse syngeneic cell line, we could able to see the FDPS fold change increases, and it increases from the androgen dependent to androgen independent condition, saying that FDPS expression could later we can correlate with the castrated resistant condition as well. We also did an in silico analysis using the cBioportal, and we found that the cases or the cohorts which has a more PETA deletion has an association of FDPS amplification as well. So our first idea is to see that whether FDPS by itself has an ability to promote the prostate cancer progression. So what we did, we took a FDPS cDNA and we overexpressed in 2 of the cell line, and we found that FDPS overexpression promotes the colony formation ability and increase the cell number from with relative to the vector control. So what could be the reason, or what are the signaling that makes this FDPS as a tumor promoter? Then we analyzed some of the signaling that is well known in the survival and proliferation, and we found that, for our own surprise, we found the increase in the phosphorylation of AKT and ERK associated with the cyclin B1 as a proliferative marker as a way for sustaining these prostate cancer cell growth. So we thought that what could be the reason for the association of this increase in the activation of this signaling and increase in FDPS level. We found that these PI3K activation, which can support AKT, and if FDPS is also overexpressed, so if it supports the prenylation process of some small GTPases, so that could able to support and sustain the AKT activation. So in this process, what we did, we analyzed some of the small GTPases, and for our own surprise, we could able to see that when we overexpress FDPS, the prenylated form of Rho-A, Rho-G, and CDC42, all small GTPases, got increased. So this shows that the FDPS modulation can be associated with the prenylation process. So as I said earlier, the zoledronic acid was found to bind or interact with the human FDPS. We did a dose-dependent analysis, and we saw that when we dose-dependently treat zoledronic acid, the cancer cells decrease the FDPS level. But just for time concern, I didn't show that data here. But here I want to emphasize that when we dose-dependently treat zoledronic acid, we could able to see an increase in the unprenylated form of Rho-A, Rho-G, and CDC42, stating that this zoledronic acid inhibits the prenylation process. At the same time, when we did knockdown of this FDPS, we could able to see a decrease in the AKT, whereas at the same time, the prenylated form of Rho-A, Rho-G, and CDC42 decreases with increase in the unprenylated form as well. This was also verified in a confocal experiment where, with relative to the control, the zoledronic acid treated cells showed an increase in the level of Rho-A and Rho-G. This is not only true with the 22R1, even the C4-2B cells did show this effect as well. So, so all these things showed that FDPS has an ability to promote prostate cancer cell growth and survival. But in terms of radiation, what we did, we chronically selected 2 of the cell line, 22RV1 LNCAP C81 cells, and we just want to see the status of FDPS. For our own surprise, when we treat these cells for 2 grays of 5 fraction, FDPS levels got increased or enriched upon this radiation treatment. And we also want to see whether it is with regard to the dose or with the dose-dependent effect. So what we did, the CE2 P10 knockout mouse syngeneic cell line, we dose-dependently treat with the various doses of radiation, and we found that FDPS increases with the increase in dose, and it correlates with the some of the resistant markers like therapy resistance markers like MDR1, ALDH1, and PAF1, etc. So we show that FDPS has an association with radiation, but functionally, what we did, we already overexpressed this FDPS in 22RV1 cells. So what we did, we equally seeded this vector and FDPS overexpressed cells, and we exposed equally them for a 4 gray of single fraction radiation. And we found that the number of surviving colonies is much more higher when we overexpress FDPS as relative to the vector control. This shows that the FDPS overexpression has an effect in protecting the radiation effect. So what we did a knockdown of this FDPS, and we found that similar trend of decrease in AKT and ERK. At the same time, we could able to see when we knock down FDPS and we synergize with the radiation, we could able to see the decrease in the number of colonies that formed after the radiation therapy. This shows that there's a reduction on FDPS as an association, or it can synergize with the radiation therapy. So having said that, the next thing what we want to do is we want to combine in the therapy way, where we pre-sensitize the cancer cells to the zoledronic acid, followed by which we expose them for radiation. As we said earlier, when we treat them with the radiation, by radiation by itself, even at the 2-gray fraction, it increases the FDPS level, but this increase in FDPS level is counteracted by treating them with zoledronic acid. So in the combination therapy, zoledronic acid plus radiation decreases the FDPS level in 2 of the human cell line and in a murine syngeneic cell line. At the same time, as I said earlier, this radiation by itself and with treatment with zoledronic acid decreases the level of phospho-AKT and ERK as well. So this shows that zoledronic has an inhibitory effect by interfering with this downstream signaling of FDPS. And we also evaluated functionally how this zoledronic acid could sensitize this human and mouse cell line. So what we did 2 different functional approach. One with the proliferation assay, where we treated them with zoledronic acid alone and radiation alone, and in combination. And we saw that a week time, we followed this for a week time, and we found that zoledronic acid mediated pre-sensitization decreases the colony growth in both human and mouse cell line, and it also decreases the colony number as well. We could able to see a moderate to severe decrease when the radiation doses increases, and this increase in the dosage synergizes with the zoledronic acid, and it reflects in the reduction in the colony growth and proliferation capacity. So overall, we want to do this in some of the preclinical models. So what we did, we adapted the mouse syngeneic cell line and tumoroids derived from these cell lines, and we found that when these mouse syngeneic cell line treated with zoledronic acid alone or radiation alone, it decreases the colony, but it is significantly decreased when you combine this zoledronic acid and radiation therapy. Similarly, the organoids derived from this beta knockout mice model showed a decrease in the average tumoroid area with relative to the radiation as well. So when we evaluated these histologically, these organoids with the pathologists, they showed that there are more number of necrotic and apoptotic cells in the combination therapy. So this shows that the decrease in the colony growth or the delay in the tumoroid growth is mainly because of increase in the necrosis or the apoptosis caused by this zoledronic acid and radiation therapy. So the overall summary, what I would like to show is FDPS is required and in the low level, and it is involved in the cholesterol and cereal biosynthesis. In case of prostate cancer, this radio selection by itself activates these mevalonate pathway genes, specifically these FDPS, and it is involved in the activation of these phosphorylation of ERK and AKT. And when you block these FDPS with zoledronic acid, it makes for the pre-sensitization of these cancer cells to the radiation therapy. And thus we identified FDPS as a novel radiosensitization target in prostate cancer therapy, and zoledronic acid as a novel sensitizer in these patients. And we want to do in conjunction with the other AR signaling blockade as well. So in future direction, we want to extend this tumor promoting function of FDPS towards the metastatic promoting function. And we want to find an association of this FDPS with various other clinical parameters like Gleason score and PSA and recurrence in response to radiation and ADT treatment. And we want to see how FDPS correlates with patient survival as well. So finally, I just want to acknowledge my mentor, Dr. Batra, for his strong support over the period of time. Because of him, today what I'm here is all because of his support. And I would like to especially thank our collaborator, Dr. Chilen, who is a strong support for me for the radiation treatment, and who teach me a lot of things for the in vivo treatment of these mice, everything. And now I am collaborating with Dr. Michael Bain for this prostate cancer work. And I would like to thank Dr. Kaustubh Datta, who is our collaborator for prostate cancer work, Dr. Leele and Dr. Tolman for pathological evaluation, and Dr. Smith for the statistical analysis. And I would like to thank all my colleagues, Dr. Murthy, Dr. Jain, Dr. Satyanarayana Rachagani, Shaktivel, and Javed, who are my colleagues who are working for the prostate cancer. And Ramakant and Pranita are the person who did all the in silico analysis. And Sushanto Halder is the graduate student who is helping me for some of the in vitro studies. And I would like to thank a special thanks for the radiation physicist, Sikong Lee and Su Wang, who always gives time for me for radiating some of my cells. And I would like to thank the UNMC flow cytometry analysis and confocal analysis. And I would like to thank the funding for Dr. Batra from NIH and DOD as well. Thank you, and I'm ready and happy to take any questions.
All right, thank you. So we're going to start the panel session, and